This video provides a comprehensive overview of the immune system. Hopefully, you will enjoy this funny journey edited by Dr. Jihad Hamid. The immune system is divided into innate and adaptive immune systems. The innate immune system is the first line of immune response. It is a fast response and acts generally, not specifically. On the other hand, the adaptive immune system has a slow immune response. It is very specific and produces long-lasting immune memory. Now let's focus on the innate immune system. The innate immune system is a highly complex integration of various components. It has three major components: the physical barriers, the cellular components, and the non-cellular components. The physical barriers include the skin with its epithelial surfaces, the gastrointestinal tract with various defense mechanisms such as peristalsis, gastric acid production, and normal flora, as well as the respiratory tract with its cilia and mucus, and many other components of the innate immune system such as tears and saliva. The cellular components of the innate immune system consist mainly of leukocytes, which are divided into either phagocytic leukocytes or non-phagocytic leukocytes. The phagocytes who are ready to eat at any time include dendritic cells, neutrophils, and macrophages. Or non-phagocytic leukocytes do not engulf the invaders. Indeed, they prefer to destroy them. The list of non-phagocytic leukocytes include natural killers, mast cells, and eosinophils. The non-cellular components of the innate immune system includes the complement system and plasma proteins. The complement system is composed of around 30 proteins that are further activated through complicated overlapping pathways. Among different functions of complement proteins are directing the phagocytic and cytotoxic cells toward the invading antigens. Some of the complement enzymes can penetrate and destroy the invader's cell walls. Here is a comprehensive view for the innate immune system. Now let's have a look at the adaptive immune system. The adaptive immune system is composed mainly of lymphocytes, B cells and T cells. Both B and T cells are produced in the bone marrow, but T cells are developed later from thymocytes. There are three types of B and T cells depending on their function: naive cells, before meeting enemies; effector cells, in the battlefield; and memory cells, which produce the long-lasting memory. Additionally, the adaptive immune system can be divided into either humoral immunity, which is mainly antibody mediated, or cell-mediated immunity. The antibodies are produced by B cells, whereas cellular immunity is mediated mainly by T cells. Here is a comprehensive view for the adaptive immune system. Now we will go deeper into the innate immune system. Every bacteria or virus invading our bodies has its own specific molecules called pathogen-associated molecular patterns, abbreviated as PAMPs. This PAMP molecule could be a polysaccharide, peptidoglycan, or even an RNA segment. On the other side, the phagocytic cells, such as macrophages and dendritic cells, have their own screening receptors for these PAMPs. These receptors are called toll-like receptors, abbreviated as TLRs. Once the PAMP molecule comes into direct contact with the toll-like receptor, it activates it. This activation of toll-like receptors results in a cascade of intracellular reactions, which end with the production of various numbers of immunological and inflammatory mediators. These mediators include interleukin-1, IL-1, tumor necrosis factor alpha, TNF-A, interferons, histamine, prostaglandin, leukotrienes, and last but not least, nitric oxide, NO. These structurally different mediators trigger a wide range of immunological and inflammatory cascades. Now we will focus on the inflammatory reactions in response to the invaders. The inflammatory mediators produced by macrophages, such as histamine, leukotriene, prostaglandins, and nitric oxide, produce significant changes in the blood vessels and surrounding nerves. They induce vasodilation, which gives rise to redness and heat, the two cardinal signs of inflammation. Additionally, these mediators increase the permeability of nearby blood vessels, which results in leakage of fluid and inflammatory cells to the site of infection. The resulting swelling is the third cardinal sign of inflammation. The inflammatory mediators also make the nerves more sensitive to pain, which represents the fourth cardinal sign of inflammation. Now let's focus on one of the main innate immune system cellular reactions, which is phagocytosis. Firstly, the inflammatory mediators stimulate the endothelium to exhibit the adhesion molecules. Then, the neutrophils are attracted and come in contact with the active endothelial adhesive molecules. This contact results in activation of neutrophils. The active neutrophils are adhered to the endothelium. This is followed by sliding of the whole neutrophil cells through narrow endothelium gaps. This sliding transfers the whole neutrophils outside the blood vessels. Then, the extravascular neutrophils start to phagocytose the invading microbes. Then, they digest and degrade the invaders. It is important to know that the neutrophils are usually the first arrivals to the sites of microbe invasion. In fact, they are the predominant cells in pus. The neutrophil's phagocytosis step is boosted by an invasion of huge amounts of other phagocytic cells, such as macrophages, dendritic cells, and B cells. These cells engulf and digest the invaders. Then, the phagocytes present some segments of the invading antigens on their cell surfaces. Therefore, they are called antigen-presenting cells, APCs. The digested microbial antigens are presented on the phagocytic cell membrane by the help of major histocompatibility complex molecules, abbreviated as MHC molecules. MHC molecules are glycoproteins that are encoded by polygenic and highly polymorphic MHC genes. In humans, MHC molecules are called human leukocytic antigens, abbreviated as HLA. In fact, the presenting of invaders' antigens on the HLA molecules of the APCs is one of the main bridges between the innate and adaptive immune systems. After we had a general look into the innate immune system, let's now go to the adaptive immune system. The T cells, produced in the bone marrow and matured in the thymus gland, regularly check all MHC molecules in our body. And they become suspicious when they come in contact with foreign bodies attached to MHC molecules. And because the adaptive system is a very specific system, as we mentioned in the beginning. There are two types of MHC molecules which bind to two different types of T cells. The first one is MHC1, which presents intracellular invading pathogens such as viruses. These MHC1 molecules bind to T cytotoxic cells known as CD8 cells. The other type is MHC2 molecules, which presents particles of extracellular invading microbes such as bacteria. MHC2 molecules bind to T helper cells known as CD4 cells. Now let's focus on T helper CD4 cells, the antibody production line. Remember, CD4 cells are the main target of HIV. Once T helper CD4 cells come in contact with foreign antigens attached to MHC2 molecules, they become activated. This activation results in the production and release of interleukin-2 cytokines. The released interleukin-2 cytokines come back in autocrine fashion to activate their releasing CD4 cells. This activation induces a cascade of intracellular reactions, ending with the replication of T helper cells to two new T cell lines, T helper 2 CD4 cells and T helper 1 CD4 cells. T helper 2 cells are the main line for antibody immunity, which is called humoral immunity, whereas T helper 1 cells are actively involved in cell-mediated immunity. Remember, interleukin-2 production, receptors, and intracellular effects are important targets for immunosuppressant drugs. Now, let's focus on antibody-mediated immunity. And later on, we will come back to T helper 1 cell-mediated immunity. Once T helper 2 cells come in contact with the B cell, it produces and releases interleukin-4 cytokines, which in turn bind to and activate the B cell. The active B cell proliferates to two B cells. Then, the newly proliferated B cells are differentiated into two functionally different cell lines, the plasma cells and memory cells. The plasma cells produces and releases various types of antibodies, whereas memory cells keep record of all foreign molecules that have entered our body to put them on the blacklist. Now we will have a close look to the general structure of the antibodies. The antibody is generally consisted of the heavy chain and the light chain. The heavy chain is always the constant region, whereas light chains are composed of both constant and variable regions. The antigen binding sites are present in the light region, whereas the Fc region, which binds to immune cells receptors, are present in the heavy chain region. The antibody can be a single molecule, such as IgD, IgE, and IgG. Or they can be a couple of molecules connected together, such as IgA antibody, or a cluster of 5 antibody molecules connected together, such as IgM antibody. Now we will have examples of antibody functions. Firstly, agglutination. Here, the antibodies form a large complex with bacteria. This complex neutralizes the bacteria and facilitates their removal by phagocytic cells. Secondly, antibody-mediated phagocytosis. The binding of antigens to the antibody activates the complement system linked to the antibodies. The complement system in turn works as a navigator for the phagocytic cells to the site of the antigen-antibody complex. Complement proteins activate the phagocytes, which become eager to engulf the antigen-antibody complex and remove them from the blood. Thirdly, antibody-dependent cellular toxicity, where antibodies attack antigens on the surfaces of the targeted cells. Then, the Fc part of the antibodies binds to specific receptors on the natural killer cells. The natural killers do not phagocytose the antigen-antibody complex, but instead they release a group of perforating enzymes which ultimately destroy the target cells. Now come back to the other branch of activated CD4 cells, which is T helper 1 cells. In this pathway, the T helper 1 cells activate the macrophages through production and release of a group of cytokines. These cytokines include interferon gamma, tumor necrosis factor alpha, and interleukin 2. The released cytokines provoke the macrophages to destroy the engulfed microbes. Tired, right? Don't worry, we are about to finish. Just one more minute. As we reach the last part of the innate immune system's cytotoxic CD8 cells, the secret world in the immune system is proliferation, and as expected, the binding of MHC1 molecules to CD8 cells activate them and stimulates them for further and further proliferation to more CD8 toxic T cells. The newly proliferated CD8 cells search the body for certain MHC1 molecules on the surface of the antigen-presenting cells. Bear in mind that MHC1 molecules represent the antigen of intracellular pathogens such as invading viruses. Once the CD8 cells find their MHC1 targets, they produce and release cytotoxic enzymes, which attack and destroy the target cells. We are almost finished, but let's sum up the adaptive immune system. This video is narrated by Hassan Nawab, medical student at King's College London.